welcome to ask questions, but please wait until the end of the lecture. In that way, I can hand you this microphone so that your questions can be recorded as well as the answers for those watching this later on YouTube or on our website. And now I'd like to welcome Hedhammar, who's here to talk about the development of artificial spiderweb. Welcome.
Thank you. And can you hear me now? Yes. Good. Nice that you decided to spend this lunch with me here. I will tell you a bit about our method to culture cells in 3 dimensions, which allows us to build tissue pieces in the lab. And this we hope to be able to use as an alternative to animal testing, for example. So I will start by telling you a bit about spiders and silk. I will not show any real spiders for those who are afraid of it, because I focus on the silk. But spiders are one of the oldest still living organisms in the world. They have been around for 400 million years. Maybe because they have this ability to make silk, I don't know, but they have been around for a long time. And some spiders can actually make 7 different types of silk with a little bit of different properties. Some they use to wrap in their eggs to protect them, and some they use to catch prey. The most famous silk is the dragline silk, which is the one that they make this framework of the nets. And that's also if you have seen a spider flee, then it uses this dragline silk as a lifeline. But all of these types of silks are made of proteins. And it's special proteins that they have in glands in the back of their in the back of the body. And then they extrude it as silk. So it comes from the back, not as the Spider-Man from here, but from the back. So this type, it's not just any type of protein that can make silk. There are special proteins. And these proteins then are stored in these glands. And they are stored as a very concentrated protein solution with a protein that is very unstructured, random. And then these proteins have this ability to reorganize and pack themselves very tightly so that a solution is transformed into a material. And in this case, then a very elastic and strong material. And that is thanks to that the protein can find each other and connect with each other in a very organized way. So that it becomes a material. So this is a very interesting property to transform from a solution to a material that we wanted to investigate more. So we have looked at these proteins, and if we zoom into a typical spider silk protein, it looks something like this. So in the end of the proteins, there are globular domains, and then in the middle, there are repetitive regions that are not exactly known how it looks yet, but a long part is very much repeated the same. So we have looked at this protein and the different parts separately to try to figure out what is it that makes this protein able to make a strong and elastic material. So first we took this very small globular end in the C-terminal of the protein. And actually this small part can also have this ability to pack the chains together to make silk-like material that looks like this. But these are not that stable. You can dissolve them if you have the denaturing agents. But if you put that part together with a piece of this repetitive part of the protein, then the protein has a very high propensity to assemble into a silk-like material that looks like this. And this silk, this artificial silk, is very thermally stable. It is stable up to 267 degrees. It's chemically stable. You need to have neat formic acid to be able to dissolve it. So it's a very stable silk-like material. Although it's then made only of a piece of the real spider silk protein. And the spiders, they have then a third part also, this in red here, that is very conserved in all spider species. But still, obviously, we can get this silk without it. But we have looked into this red part, the N-terminal part as well, and found that it has a very interesting property. At pH 7, which is neutral pH, this part helps to keep the protein in solution. So it hinders from the silk to prematurely form fibers. So in the gland, when the protein should be stored, this part keeps the proteins from going together with each other and make silk. And then at pH 6, on the other hand, it instead makes this silk assembly even faster, so that the silk is formed instantly. And this is, of course, important for a spider, which then, for example, if it has to flee, it has to make silk like this, not in minutes. But for us in the lab, we only use this part to facilitate the production of the protein, to keep the protein soluble, but then we actually remove it in the final part, because this part is the only part that is not totally compatible with our body. So we have decided to not use it because we don't need this instant formation. But this part then, the 10% end of the silk protein, that is then have this propensity to make a silk material, and it is still small enough to produce in systems that are easier than spiders. So we have chosen to take this part of the protein and produce it with a recombinant system. That means that we have taken this gene and taken it from the spider and put it in a lab bacteria. And this lab bacteria we can cultivate in bioreactors. So we get a lot of them and then tell them to produce a lot of proteins. There are seeds over here if you want to. So we can get a lot of the silk protein and purify it with a controlled process so that we get a pure protein of only this spider silk protein in a controlled way. And then this part of the silk protein then, we have looked a bit more into how does it transform from a protein solution to a silk material. And it turns out then that this protein has the propensity to rearrange into the silk material if it gets to be at an interface. And this interface could, example, be water-air interface, a very simple interface. So the C-terminal part of the protein, it has a very strong ability to go to the air-water interface. And then it brings also the rest of the protein, and there along this air-water interface, the protein get help to align, and then they can rearrange and pack together with each other and form this silk material. So by just this simple help of an air-water interface, the proteins can transform into silk-like materials. So this, to utilize the air-water interface, is a very mild way to produce a material. And you can also quite easily adapt it depending on what type of material you want to have. So this, to have the silk protein and air-water interface, there you can play around a lot. It's actually very fun to play with this protein. You can do a lot of things. If you, for example, just keep the protein solution standing still, then the proteins will assemble and form a transparent film. If you have this air-liquid interface expanding and relaxing back and forth, then many smaller fibers in a bundle are formed. And if you have the liquid, the silk solution liquid, and introduce air bubbles into the liquid, then you get a foam-like, net-like structure. And there are also other things that you could make by playing with the silk protein solution and just air liquid interface. So it's this that you see on top here, the network here. That is what got me to think of tissue engineering applications. I don't know how many of you that have ever thought about why are our body functional? What is this built up? Why are we not just a pile of cells? But what is it that keeps ourselves together in a ordered way and makes us have functional tissue? That is things that I think of a lot. And so if you zoom into a tissue, ordinary connective tissue, it actually looks something like this. So the cells in our body are growing in 3 dimensions. And they can do that thanks to that they have like a framework to grow on. And this is the thing that you see between the cells here. And that is called the extracellular matrix. So it's like a network that the cells can adhere to and grow and climb along. That's what keeps our cells together into tissue. Since about the 1950s, people have been able to culture human cells in the lab. And then typically people use these type of plates, tissue plates, a petri dish or something, where they culture the cells. The cells in a dish like this are growing in 2 dimensions. They have mostly contact with a hard, flat surface. And then on the upper side of the cell, it's media. So it's actually very unnatural to grow along something hard and flat and not be in connection with each other. And in the body, the cells are growing in this extracellular matrix, which is then more like a network where the cells have more contact with each other and with the matrix, and not are forced against something flat and hard with the upper part exposed to just liquid. So something like this would be much more biologically relevant for a cell. So in order to be able to allow a cell to grow in 3 dimensions, you need to have something to support it. You need to have something that mimics how it is in the real body, some mimic of the extracellular matrix. And it should then be some kind of framework that is strong and elastic so that the cells can sense it. And it should also still be thin enough so that the cells have more contact with each other rather than a material. And if the cells then get in such an environment, they will get the signal that they are in the right place, and they will produce their own extracellular matrix. And then it's of course good that the framework they had from the beginning is degraded and replaced by their own natural extracellular matrix. So these first things here, strong, elastic, and thin, that's exactly spider silk, right? And spider silk is also degradable. In the real extracellular matrix in our body, this network also has some bioactive modules. And that is pieces of protein that tells the cells to adhere and tells the cells what to become and how to grow. And these bioactive things are, of course, not present in natural spider silk because that's not why the spiders are producing their silk. So that is the only thing that is lacking in the spider silk. But with our recombinant DNA technology, we can fix that. So what we have done is that we have introduced bioactive modules into our silk. And this we do by combining the gene that encodes this silk protein with the gene that encodes this type of bioactive modules. And then we'll let the bacteria produce this protein that is then a combination of the silk and the bioactive modules. And we can do that in a similar process as for the silk. So we get then a silk protein that is fused to, that is decorated, that's linked to a bioactive module. So this propensity of the silk protein to form a silk material, that is actually maintained even if it's attached to something else. So the silk part of the protein still can interact and rearrange and pack together to make a silk material. And since this happens then at this air-water interface, which is a very mild condition, this bioactive module is also kept in a functional way. And it is attached then to the silk material and exposed outward and with a maintained bioactivity. And this is what we call a biosilk, which means then a silk with some bioactive module. When a cell adheres to extracellular matrix, they do that with some receptors in the cell membranes that bind to the extracellular matrix. And then the cells get the signal that they are in the right place and continue to grow. So this is actually also what happens when a cell is put on this biosilk. So on the biosilk, the cells, the receptors of the cells can bind to the bioactive modules in the silk and get the signal that they are in a natural environment and continue to grow. So here is an example of a human cell that is on a biosilk, and that we have stained here the interaction between the cell receptors and the material in green. So you can see here that they are attaching to the biosilk. With this bioactive part and the three-dimensional network from the silk, we think that we can build tissue. But how should we get the cells into the silk? Well, since the silk is then transforming, the silk protein is transforming to the silk under these mild conditions as the air-water interface, we thought that we could maybe do this together with cells. So we have developed a protocol where we have a silk protein with the bioactive module. And then to this protein, we add cells in cell media in a culture dish. And then we introduce air bubbles to get these interfaces to transform the silk. So we take a pipette tip and pipe it in air bubbles for 5 to 10 seconds. And thereby we get a three-dimensional foam with cells in. And then we'll let this stabilize for about 15 minutes, and then we can add more cell media. And this is what we get. Here you can see, I hope, some white things, which are the silk network. And in between them, there are cells. We have here stained the cytoskeleton of the cells in green and the nuclei of the cells in blue. So the cells get integrated in this network of biosilk. And this procedure then to just pipe it in air bubbles for a while is mild, but we thought we should have anyway check that it doesn't harm the cells. So we looked at the viability of these cells in these three-dimensional networks, and we have a very good viability actually for the cell types that we have tested, and we have also applied this very same method for also stem cells, and they also survive this mild procedure. So this is very encouraging. So we thought that we should compare this to other nearest technologies that exist. I don't know if any of you have heard of this bioprinting, things that you can have cells in gel and print it to various three-dimensional structures. Yeah, really cool, yeah. And we thought that we should compare this to this, because here we also get something in 3D. So we decided to do parallel comparative studies where we have cells in our biosilk and compared with cells in one of the most common of these type of hydrogels. And then we chose a hydrogel that also have this bioactive module in it. So it's the same bioactive module. And a little bit to our surprise, it's very different results. The upper curve here is where the growth curve for cells grown in Biosilk, and the lower one is the growth curve in this hydrogel. And these pictures are live dead stainings of cells after 2 weeks in the Biosilk above and hydrogel below. And this is quite typical actually to what you see that people publish about cells in hydrogels, that the cells stay viable very good. They are viable, but they don't expand, they don't grow. So they are kept viable, but they are not growing. While in the silk, the cells are expanding and fill up the space and become something that looks quite like tissue. So we wanted to understand what is the reason behind the difference here. So we zoomed in at early time points and later time points to see what is happening with the cells in these different environments. And what we see is that in the Biosilk, the cells start to adhere and spread out already after 1 hour. And then they continue to grow and fill up the spaces in the Biosilk. While in the hydrogel, the cells are encapsulated as round cells. And they stay as round cells throughout the period. They are viable, but not much more is happening. And we speculate that this is that in the Biosilk, the receptors on the cell membrane have something that is similar dimension as the real extracellular matrix to grab on and gather several receptors and grab on and get the signal, hey, I'm in the right place, I should grow here and expand. While in the hydrogel, there's a gel with one molecular thick things. So it's not enough room or strength for a receptor to actually grab onto and spread out. So it's a little bit as it's possible to climb in some kind of framework, but if you're just swimming in a pool of gel, it's difficult to move around. That's how we think it is. So we think that we have something here, the Biosilk framework, that is a good start for growing cells in 3 dimensions into tissue. And this we have now taken several projects about to try to make further into reality. And I will tell you about some of these efforts. And I will start by telling a bit about how we try to make skin using this Biosilk. So skin is an example of an organ that would be very good to have an engineered variant of, because then you can do different types of tests of toxicity or drug delivery, etc. But the skin is also our largest organ, so it's to be able to make a relevant test, it would be good to have bigger pieces of this tissue. And if you have a bigger tissue, then you need vascularization in this tissue, because all cells need nutrients and oxygen. So there we have some challenges. The skin is also composed of 2 layers of different cell types, which we would then need to be able to combine. So the first challenge here, as I described, our first method of having the cells in a 3D network was by pipetting air bubbles. If you should do that to something larger, you will get very tired in your jump. I can see who is working in the lab here who understands. So what we did then was to try instead with a latte whisker from Kjell & Company. So you can take this latte whisker in the protein solution and put it on for 5 seconds, and you get a foam. And then this stabilizes to look like this. And we have actually viable cells throughout this bigger chunk. And in order to get this piece also vascularized, we want to have some microvessels in this. So to do that, we have taken a portion of the cell type that makes microvessels, the endothelial cells, and blended that in together with the skin cells, and then made this procedure. And since this network of the silk fibers is flexible and it's quite much room for the cells to move around, it turns out that the microvessel forming cells are finding each other in there and go together to make this type of structure. We can see then these type of structures here. The microvessel cells, the endothelial cells are stained in red. To be able to see that this is actually not just branches of cells, but actually some kind of with something inside, we made sections like slicing it and looked at consecutive section, and you can see there that it's actually also a lumen in these. So they can communicate with each other and arrange themselves into something that looks like microvessels. So this first part, this is how it looks, a real photo of first part of the skin that we make. And then we put the ring around it, and then we seed the next type of skin cells on top of this, because to get this bilayered skin. And then we get something like this. This is a section of a skin piece that we made at the lab. So you see the bottom part is similar to the lower part of the skin, the dermis part. And then above we have this outer layer of the skin, which has then cells that produce keratin. That is the thing that is on the outside of our skin. And when you touch and feel these small skin pieces, they are actually feeling a bit like skin. They are keeping together, they have the mechanical properties similar to real skin. And this is what is different compared to if you have them just in a hydrogel. The cells are actually connecting with each other, and you have something that have a morphology and mechanics that is similar to real skin. And this is important if you want to have a model when you study how things penetrate through the skin. It's important that the skin has this right morphology and not just a gel where anything can pass by. So we hope that this could be a good thing for studies of skin in the lab. Other types of tissue would also be good to have as models in the lab. For example, it would be good to be able to have models of tissue pieces that you could test new drug candidates on. Typically today, the initial tests of a drug candidate is to have cells cultured in this 2D system on a Petri dish. And as you have seen now, it's not that biologically relevant to have cells like this. When they have half of the surface exposed to the media, they are very sensitive. So it's quite many substances that could be effective on these type of cells since they are so sensitive. So it has turned out that it's only 10% of those that work on cells on 2D that then actually works in the real situation in vivo. So it's, you get quite many false results by testing on cells in 2D. And it's quite logical. In animal models, of course, in animals the cells are growing in 3D. So there the cells are in a normal way. But with animal models there are of course some other drawbacks. It's the ethical issue, and also that it's animal cells and not human cells. And if we want to have drugs for humans, that matters. So to be able to have human cells cultured in a in vivo-like environment in 3D would be a better representative for a lot of tests. For example, one cell type that is very dependent on how the environment is cancer cells. Cancer cells grown in 2D are not staying, they are affected by that, they're not staying as they should, so you don't get representative results by having the cancer cells in 2D. Because in a tumor, the cancer cells are growing in 3D, and they have extracellular matrix, and they also have normal cells around them. That's the environment of a tumor. So it would be better model for a tumor if you could have a 3D environment and also combine the tumor cells with normal cells. When a patient is diagnosed with breast cancer, the tumor is removed and used for a diagnosis. We have a collaboration with the Bröstcancercentrum at KI, and with help from them, when they take out the tumor, they can just gently scrape to get a few cells without destroying the diagnosis of the tumor. We're just taking a few cells. And those cells we have tried to integrate in our Biosilk to make a replica of the tumor. So this can then be done without disturbing the normal procedure. And here is an example of then these few cells taken to integrate with our Biosilk, and the cancer cells then form a 3D tumor, and they also maintain these typical cancer marker, so they look as if they were still in the real tumor. So my dream and our aim is to be able to use this method and develop it further so that we can take a few cells from patients, make many small replicas of the tumors, have them in the lab to be able to test which treatment would be best for this patient. So since we can take a very few cells and get these type of mini tumors, they form, we get these tumors in the lab within 2 days, and then you can do testing within a week. And then you get these small replicas of the real tumor where you can see at cellular level which drug is most effective against the bad cancer cells without harming the good cells. To select a method, a treatment option that would be effective for the patient, but still as little side effect as possible. And to be able to do this quickly and to know how to kill those of the cancer cells that are the ones that otherwise would form metastasis. Because it's very important to capture those and kill those early to avoid spreading. This is one of the things that we are working on now that we hope this could be useful for, since we can get a very efficient integration. We can use a small sample of cells to have many replicas to try on. We also want to make more advanced cancer models, because for studies of how new cancer treatments could work, it would be good to have better cancer models to be able to follow what is happening. And then it would be good to have a tumor in the surrounding tissue, and preferably also be able to see how is the cells communicating with each other and also with the blood system. Especially for the newest type of cancer treatments, immunotherapy, it would be very good to be able to see how something goes from the blood system into the tissue and how it affects the cells. So we are trying to make a model where we also have a bigger blood vessel to be able to administrate drugs through. Blood vessels are quite complicated to imitate. The base of a blood vessel is a thin, permeable membrane with two different cell types on each side. And together with a group at the Microsystem Technology here at KTH, we are trying to go for this. So we get help from them to construct a special culture well with a frame that can support a blood vessel. And on this frame, we make a thin silk membrane that is less than half a micrometer. Still, this silk membrane is strong enough to be able to handle, and it also has a very good elasticity. And it's permeable for the proteins that are in the blood, that are important to be able to pass through the blood vessel. And since it is made of Biosilk, you can grow both types of cells that are in the blood vessel on each side. And these cell types then form a layer that looks like the inside of a blood vessel. So here we are now, and our next step is to have this blood vessel mimic embedded in a tissue. And the tissue then we will make in a similar way as I showed that we made the skin of, to have a surrounding tissue around this also. So that's our strategy to also make more advanced models, getting closer to how it is in a whole system. So I just want to end up with showing some highlights of what it is with the Biosilk and why it could be used for making tissue models. So these silk proteins, they have this ability to form a silk material at the air-water interface, which is then a very mild condition. And there they make this strong and elastic fibers that are similar to the extracellular matrix that are naturally surrounding the cells. And thanks to the recombinant technology, we can also put in the bioactive modules that gives the right signal to the cells. And in this, the cells can adhere and get the signal that they are in the right place and continue to grow into a tissue. And that they also can have room to migrate and connect with each other and make a tissue. So we hope that this method is something that we can use for a lot of different things to help other researchers move on with different models and understand better, and yeah, do a lot of good things with. And this is the people that makes this possible. We are really doing our best to develop this and understand it better, and also collaborate with others so that others can take it forward to certain directions. If any of you have any ideas, you can just contact us and we will try to do something. We think it's necessary to have a lot of collaborations to be able to use it for different things. We also, of course, these are the base for having this done. Fantastic people, and we are of course very thankful for support from Vetenskapsrådet, Vinnova, Knuten Alice Wallenberg, KTH, and the company Biotechnology that produces the silk proteins. If some of you think that this with alternatives to animal testing is interesting, I want to recommend that you come back next Thursday. There will be a seminar here where 5 different researchers will discuss different options for having alternatives to animal testing. Thanks a lot for listening.
Thank you. does anyone have any questions? There we go.
Hello, thank you for the talk. I think it was very inspiring and good. But I have a couple of questions. I'm working also with 3D models with collagen, fibrin, and other hydrogels. But most of the time, why we are using is because the cells can degrade that thing, like the mattress. But according to your picture and the talk, I just think that how they can remodel the silk or how they can degrade the silk. Because sometimes for cancer cells, they have to produce their own ECM. While they're producing their own ECM, they have to degrade the air surrounding. So is it possible that they can degrade your silk?
Yeah, and it actually promotes production of own extracellular matrix that we have seen. So they are filling up with their own extracellular matrix quite soon. In vitro, I don't know how much of the silk is degraded. I know that it's degraded in vivo.
Okay.
When we start, it's quite much room left for cells and a new extracellular matrix. So in an in vitro culture, there is plenty of room for expanding of the cells and real extracellular matrix. But we haven't been able to see how fast it degrades in vitro, but we know that it degrades, depending on where you place it, it degrades in vivo after 1 to 3 months.
Okay. Can I ask another question also? Yeah. Also, I was thinking that can you play with the softness or stiffness of the silk to mimic it? For example, like some cancer types love stiff environment, and some cancers prefer being in a soft 3D mattress. So can you make it like a thicker silk and different alignment of the silk so they can make it much more stiff environment? Because the picture you showed with the allogenate, actually it's because of they're in the stiff environment, so they don't go around that much. They don't adhere to the 3D.
Yeah, we haven't looked that much at making different stiffnesses, but the density of the silk you can vary quite easily. And I didn't show any example here, but we also can make the cells in when the fibers are more lined, then you get another mechanics of it. And for example, in order to mimic bone tissue, you can have in minerals to make it stiffer. But that's definitely interesting to look into.
I think we have time for one more question. No one? Oh.
Is there a preferred spider species for this kind of silk, or would any spider do?
I actually think that any would do. Okay. We have chosen, we started with one spider that is making silk in the water. So this silk is, we know from the beginning that it's good to have in water, and it has this repetitive part of the protein is in a way so that it easily forms silk. So we might have been lucky to have chosen a good one, but I mean, you can vary the protein sequence quite a lot. Okay. So.
Lovely. So thank you very much for joining us today. Here's a little
thank you.
gift from the library. And to the rest of you, there's going to be another popular science lecture on December 4th. We can hear Kjetil Falkenberg Hansen talk about the technological and musical development in hip-hop. But thank you very much for coming today, and thank you, Mi.
Thank you.